one video take one the other thing and then tinenya galundo ke video dadin and video gurane video you check on a weight about the properties of enzyme ke characteristics of enzyme gurane ke video idan as a reminder check on a lot of check on enzyme check on kada ka other substance because check on a lot of ka protein can enzyme ideal and also check on a lot of their biological catalysts that like an reaction like an speed up and check on a lot they are also specific which means like an involve reaction killing karade and check affect a reaction more and check on a lot no like affect a pH temperature and also other substances that act as inhibitors and today I'll touch upon week quality and the mabana wale a short kind of classification of enzyme enzyme la kon ko dagade keno ko kon ga classify ki chodin ko kon chagade ki chodin jagde de tuma kon e ko kon ko wala and title ka title and to how do we name and classify enzyme how enzyme chalka kede ki ko ka classified common or working enzyme nomenclature naming of enzyme ki ta kwa enzyme kwa chodin kwa ka jagade then we want to go wale as a point and table 3.1 gives some example of enzymes and the reactions they catalyze so the champi ke chod enzyme trick reaction ta ko kana catalyze ke reaction ko kana involved ke reaction ko kana speed up in we can describe that in many ways and I can take a quick video can banner with out a table animal so then lipase enzyme just coming on a hydrolysis of lipids so we're going to come fatty acid can glycerol ATP hydrolysis of ATP and we can also add sucrase enzyme there on madguambo hydrolysis of sucrose and succinate dehydrogenase so kachwedema dehydrogenase luno D makwelo matatana one bar remove dehydrogenase luno removal of hydrogen ion or find an hydrogen atom from succinate or during respiration by hydrogen by remove guama in this reaction and DNA polymerase joining of nucleotides to form DNA or deoxyribose nucleic acid so a nucleic acid the nucleic acid form two strands of nucleotide and it's coming on a nucleic acid nucleotides that are building block of nucleic acids and this nucleotide can take a nitrogen base sugar can a phosphate group and that will join kadaro like in DNA like a form DNA it's a double strand whereas RNA a single strand in gretane and mandarin pepsin so pepsin is a digestive enzyme that involve digestion of protein in the stomachs of mammals in into the mammal gland takuchako can produce the stomach then take a enzyme which are pepsin and enzyme more did involve digestion of proteins protein digest and mandarin different enzymes are named in different ways and most commonly enzymes are named by adding ASC to the part of the name of the substrate so enzyme need one like a qua chalke ke chod medong ba substrate mutaten ba wini later the job then ba ASE ba add ke ba matan so which enzyme ka bakulungun for example lipase enzyme just ke chod de kon ngai ti ro be ka lipid and sa lipid cha I can d cha ko wood then cha ASE cha matan mulo no ko indicate no enzyme and cha mo ke ASE and enzyme moladang hydrolysis of lipids okay, in bracket come lipid hydrolysing enzyme, and sucrase enzyme called ASE, and this sucrase enzyme lademu a hydrolysis of sucrose. Sometimes the enzymes are named on the basis of the reaction that they catalyze. Enzyme reaction catalyze. For example, polymerase. So polymerase the reaction catalyze polymerization process. So polymerization process monomer units form, then become polymer, become form, and process of polymerization. Let's take monosaccharide. Monosaccharide back join as a single unit, then become polysaccharide become form, and process of polymerization, and different type of polymerase enzymes can involve reaction in me. And dehydrogenase, removal of hydrogen atoms or ions. And some enzymes have been named based on the sources from which they were first identified. And Tangwani take an enzyme to large picture for example, papain from papaya, kimulakonatural popo. So enzyme majigika popo for the first time in Daju Chakjut kojigika popo like papain. And others are named according to where they act. Acting can enzyme for example, intestinal protease acts on proteins in the intestine. So should give a quick information to what intestinal they intestine act protease they protein acts on protein in the intestine. The name of some enzymes ends with IN, indicating that they are basically protein. For example, pepsin and trypsin. IN corina can enzyme protein. And these enzymes usually have an alternative names that tells you rather more about them. For example, the alternative name for a pepsin is gastric protease. This tells you that it acts on the proteins and it does so in the stomach. Our chodati can enzyme to good IN can get chodi cocaine. So chodi and cocaine lack on a information my did. Let's say our pepsin chodamodoni a gastric protease. So gastric is something that it is found in the gastrointestinal or in the gastric. So a stomach. The stomach and mutual protease the enemy protein, and gastric protease the act on proteins in the stomach. And because of the varied way in which enzymes had been named, biologists at the Enzyme Commission decide to produce a systematic way of naming enzymes based on the way in which the enzymes act. To appreciate this, we must first look at how enzymes are classified. Enzyme in different way, just by adding ASA and sometimes reaction can catalyze and confusion let's say teramero chakchut ke enzyme ko gwada majageten ten teramero din enzyme no chakchut ke gwada way dong chechar ke de chakchut just by adding ASE then gwada right can take a confusion within with create combine kanito, especially mika biologist na kanito te Enzyme Commission a group and then chikarodi kuna machikar wato enzyme no baka chakchut way aku can act key reaction aku can catalyze baka chakchut keje so bukan ukoro dan kuliling agwa. And our Enzyme Commission gwama keyword body set up to produce a systematic way of naming enzyme. We can say it's a group and chikanoti enzyme no baka chakchut kadwa bame chikarodi link so mani de ko kemi karodi kuliling okay do mi chakchenamo. and enzyme classification and the systematic nomenclature of enzyme. Tata enzyme jakadakide can study can enzyme. Enzymes are generally classified on the basis of the type of reaction that they catalyze. 6 groups of enzyme can be recognized on this basis. So enzyme jakachakshut the reaction ko can catalyze and government jakamoika group dambakel can enzyme. Let's look at the table. Classification of enzyme, number class, and the reaction they catalyzed in the middle, and finally some examples. And manium oxidoreductase. So from the name oxidoreductase, it means that take a both oxidation reaction kind of reduction, so adding and removing something. And transfer of hydrogen and oxygen atoms, a reaction involved in a transfer of hydrogen and oxygen atoms or electrons from one substrate to another substrate. So reaction it can be either so polarity or it means that lose gain. So both losing and gaining of hydrogen oxygen can electron substrate to the another substrate. The reaction can catalyze. Example dehydrogenase dehydrogenase, meaning that a removal of hydrogen, which means the hydrogen transfer, and oxidase oxygen add removal hydrogenase a hydrogen removal of hydrogen oxidase oxygen is added. And class around a transferase transferase or transfer, right? Transferase of a specific group, it can be a phosphate or methyl etc. Okay, so phosphate group phosphate group, as we all know, nucleotide nucleic acid phosphate group, and that group transfer from one substrate to another substrate. We can to reaction to involve enzyme to transferase enzyme. Example kinase and transminase enzyme needed. And cladodian hydrolysis of a substrate, which means water is added in this reaction. So tapu add this reaction. Example digestive enzyme esterase enzyme digestive enzyme reaction involvement a hydrolysis of a substrate, and class into hydrolysis. And madodine isomerase isomerase, actually isomers similar molecular forms. They can be substance, they can be atoms, they can be elements, and molecular form method isomers. But isomerase reaction catalyze a change of the molecular form of the substrate. Example fumarase can phosphohexoisomerase enzymes. And number 5, lase enzyme, non-hydrolytic removal of a group or addition of a group to a substrate. So a group is in removal either removal or addition of non-hydrolytic group. Then non-hydrolytic methyl hydrogen, either bar remove or bar reduce, bar remove or bar add. Example decarboxylase, a carbonates carbon group within aldose aldine, this a nitrogen group nucleotide either back add. And Bacillin ligase enzymes, synthesis enzyme, in other words, joining of two molecules by the formation of a new bond. So molecule daro join new bond form form. And mostly ligase enzyme involve formation of the double strand of the DNA genetic and guala ko translation ka transcription process la kan yadatan, and that transcription process kwala dini la kwa form katan that process can kono with in detail la kan kwa involved in kan enzyme to example citric acid synthesis enzyme, take a class mutale ligase enzyme which each class of enzymes contain several different but related subclasses. Tau enzyme class na tepai kon with the oxidase transferase isomerase kan dia bakalati tau nyako dang no bo ke subgroup classes de ko kenda tekan kada but also tekan kemai ko kan chart ke each subclasses is further divided into subclasses. Tau subclass ni tepai kon kada from class na akanyo da kadang no madage. Within the subclasses, each enzyme has a number. Tau any enzyme take a shoot. So number enzyme de kon jing de kon ka subclass na ke kan number enzyme kan enzyme dia. Any enzyme take a shoot. Take a number, sorry. And manyam a class and number killing town. So manyam they going to 1 up to 6 and then they can count. So let's say we can example so in the systematic name of enzymes, an enzyme will have a name such as EC 3.4.11.1. Each part of the description tell us something about the enzyme. Kacha enzyme structure kajam jikon chola EC 3.4.11.1 take it to ka enzyme de kon ngajere how de kon ngajere. So EC stands for enzyme commission. So EC lo dane enzyme commission or that body the enzyme chikan chol kachudi, and the first name show to which of the 6 main classes the enzyme belong. So 3 ma ku indicate na number 3 ka class na te ku kwa enzyme mo kwa jiktan to kona lara ni na 3 a hydrolase. So mutawa 3 manam de kona ngai te enzyme mo take a hydrolase class. And the second figure indicates a subclass. A form ko indicate a subclass ke 3 baka nyoko dak a subclass ne tote ke hydrolase enzyme ne baka dak nyok, and then madeka number 4 mo banyogote banyogo narate enzyme take a subclass me, and the third figure give a subclass nyok mitanyo ko lele 11 ke third figure me ko manyama first figure 3 roda 4 during 11 1 and 1. So the third figure during 11 you then subclass ne 4 banyo go daganyo ke subclass ni tadi number 4 bakanyo go daganyo then 11 mi banyo go nyo dan matano. And the first figure is the serial number of the enzyme in its subclass, and the first figure ke number 1 mo ko indicate serial number of the enzyme ke enzyme de number 1. So then chodin manam pa nyo nga class, the main class ka class and kelan, and then manam k3 in this case then you know a hydrolase, and 4 ko nyo na subclass, 11 a subclass, and 1 a serial number ke number of enzyme mo titan. Enzymes EC 3.4.11.1 is a hydrolase 3. All the enzymes in the class 3 hydrolase some kind of bond. Hydrolase can involve hydrolysis of a substrate, right? And 3 no take a class hydrolase enzyme, then hydrolyze some kind of bond. And number 4 a peptidase. All the enzymes in the subclass 4 of class 3 are peptidase and hydrolase peptide bonds. Okay, so peptidase, all the enzymes in the subclass 4 class 3 which polarity on 3 main class. Then on that main class, our 4 to take a 4 so 3 baka nyo go down 1 2 3 4 something no then we take a 4 tower get a chale peptides so hydrolyze and chikon nga yo kwe hydrolyze in some kind of bond, right? Then peptides guame kwe hydrolyze in peptide bond kangala kwa form ka peptide bond sorry ka amino acid, so which means kwe amino acid kwe ka hydrolyze at this time. And guame number 3 no point 3 an aminopeptides. All the enzymes in the subclass 11 of the subclass 4 are aminopeptides. They hydrolyze peptide bond at the amino end of the polypeptide chain. So tanya go ka subclass 11 guame majero da ka subclass 4 tanya go subclass 4 baka nyago da enyok build ka welkel and welkel yo guame chale amino peptides. So aminopeptides among they hydrolyze peptide bond at the amino end of polypeptide chain. So amino acid la kwa form la kwa gantika bond jale peptide bond, and at the end of that chain then reaction la kwa occurs la kwa hydrolyze can peptide bond. And Madarin point number 4 okay first figure maluno number 1 tongmi lucile aminopeptides. This particularly aminopeptides is the number 1 of this subclass. So enzyme में chale lucile aminopeptide is देखो ना मैं क्या गोरो बाटी दियल बाका kon with non-concordant classification. Then finally we conclude enzyme ka first figure and then I download the classification. This is all about today's lesson, and we take a teach we take a makana loidon ke we don't take me talat jack the lara and we take we don't take yana the table and quite like then quite share yeah jack Ramadan and to line the table kaya na ida kon to subscribe ke kuyan ka like and mati ga madi martin de yanalar, and this is all about today's topic and time was